Hello students, I am Dr. Salahuddin, working as Associate Professor, Noida Institute of Engineering and Technology, Pharmacy Institute, Greater Noida. Today, I will discuss about the physicochemical parameters used in quantitative structure activity relationship. So, in this lecture, I will discuss in detail about the partition coefficient. This property used in the quantitative structure activity relationship. Now, the physicochemical properties. So, before starting the physicochemical properties used in quantitative structure activity relationship, I will discuss about the QSAR, the definition of the QSAR. So, as the QSAR says, it is a correlation between the biological activity of a series of compounds with the chemical compound and to their physicochemical parameters in a quantitative fashion using a mathematical formula. So, in this lecture I will discuss about the various types of formula, the various formula used in the physicochemical parameters in the QSAR. So, QSAR is what? QSAR is the ligand based approach used in the drug design and the quantitative measurement of the biological and the physicochemical properties of the molecule. So, here, physicochemical properties, what is the influence of these physicochemical parameters with the biological activity? How it affects the biological activity? If I am changing from one molecule to the other by changing one substituents to the other, so the property of the molecule will change, because the one substituents have all the substituents have different types of characteristics. So, if I am changing one substituents with the others, so the whole property of the molecule will change. So, here we will discuss about the influence of the different physical and the chemical characteristics of the compounds, or we can say the ligand, on the biomolecular with which it interacts, connected to its capacity to elicit the pharmacological action. Now, here the physicochemical parameters involved in the compound. There are so many physicochemical parameters involved in the compounds involved, but here in the QSAR we are taking the some very important physicochemical properties involved in it, like hydrophobicity of the molecule. Next is hydrophobicity of the substituents. Why we are taking the hydrophobicity of the molecule? So, here the hydrophobicity of the molecule, with the help of we can observe the partition coefficient in terms of the log p. Next is hydrophobicity of the substituents. If I am changing the substituents, so every substituents have different characteristics. In terms of, here I am talking about in terms of hydrophobicity. So, the hydrophobicity of the substituents shows a different characteristics in whole molecule. Next is electronic parameters, electronic properties of the substituents. Next is steric properties related with the size, shape and the hydrolysis of the molecule. So, these are the most common properties used in quantitative structure activity relationship. So, first thing I will discuss here in detail about the partition coefficient. Next is the partition coefficient is one of the most important parameters used to influence the drug transport and the drug distribution. As here the definition says of the partition coefficient, it is the distribution of the drug molecule in the octanol water system. It can be measured. The hydrophobic characteristics of the drug can be measured experimentally distributing the concentration on the relative distribution in octanol and water mixture. Next thing is hydrophobic molecule. So, here the hydrophobic molecule means water hating molecule will prefer to dissolve the octanol system, not in the layer of water, because it is a hydrophobic molecule. So, if the molecule is hydrophilic, it will prefer to dissolve in the water system, because it is a hydrophilic molecule. So, we can observe the hydrophobic characteristics or with the of the molecule by taking these two layer system, where the distribution of the hydrophilic molecules over the hydrophilic molecules and the hydrophobic molecule. Now, next thing is physicochemical in the partition coefficient, the formula says it is a concentration of the drug in octanol and the concentration of the drug in aqueous system. So, that the partition coefficient is the ratio of the concentration of the drug in the octanol with the concentration of the drug in the aqueous solution. So, here the value of P, it may be more, it may be less. So, if it is more, it is high P value, means hydrophobic molecule, or it may be less, this low P value, that is hydrophilic compounds. So, how we can produce the hydrophobic compounds and the hydrophilic compounds? We can produce the hydrophobic compounds and the hydrophilic compounds by varying the substituents. By varying the substituents, we can change the substituents on the lead compounds on the same series or the same analogues having different hydrophobicities, and therefore, we can produce a different P values. Now, we can correlate the biological activity with these P values and how it affects the biological activity by changing the P values. Now, here there is a one formula arises that log 1 by C, that is K1 log P plus K2. So, the log range of the log P is restricted with a small range, that is 1 to 4. Why 1 to 4? If 1 to 4 means if it is less, that I have already discussed in the previous slide, that if the log P value is less, it is more towards hydrophilic. Now, the activity of the drug often related with the P. I am taking here, there is a one example of the molecule by binding of the drugs with the serum albumin. Here we are taking a series of the compounds and we will observe the graph between the log 1 by C and the log P. These are the compounds. The blue color dots shows the compounds, and the log P value of the compounds increases So, here there is a one straight line graph as the log 1 by C also is greater than log 1 by C means the biological activity also increases. As here on the basis of this graph we can say if the binding increases our log P increases. So, here binding is greater for the hydrophobic drugs. Now, come to the next, the straight line relation between the log P and the biological activity is observed in many QSAR studies, because the long range of log P values study is often relatively narrow, as we can see that the log P value, that is, it ranges between 1 to 4, and there is a number of the substituents that we can produce by with the help of substitution and producing a same series. So, the log P ranges between 1 to 4. So, the range between the two values will be less. For example, the study carried out on serum albumin binding was restricted of the compound having log P values in the range between 0.78 to 3.82. There is a limitation for the drugs which binds to the serum albumin. Now, the partition coefficient, this is the very important characteristics that that influences the biological activity. So, here in some cases the parabolic curve obtained. So, in the cases where the parabolic graph obtained, so in that case the biological activity of the compounds, that is log 1 by C equals to minus k 1 log P square plus k 2 log P plus k 3. K 1, k 2 and the k 3, that we can observe these values with the help of statistical analysis. When the P is small, the log P square term is very small and the equation is dominated by log P term. Whereas, the relativity of the few drugs, that we can observe the relativity of the drug molecule with the log P when the compounds of that we are observing in the same series. Okay. And the operate in the cell membrane where hydrophobicity is the dominant feature controlling their action. So, this partition coefficient is very important step, or the very important physicochemical properties is one of the physicochemical properties to predict the biological activity. In the QSAR, as I have already said, that we cannot take all the parameters in our study. We have to choose some more very important characteristics that are involved in the biological activity. So, that here I am discussing about the partition coefficient. So, here another example, general anesthetic activity of a range of ethers is found to fit the parabolic equation, as I have already said that log 1 by C equals to 0.22 log P square 1.04 log P plus 2.16. So, how what this formula says? We can observe the anesthetic activity with this formula if anesthetic activity increases with the increasing hydrophobicity. Increasing hydrophobicity, how we can increase the hydrophobicity of one molecule to the other molecule? It is very simple. We can substitute with the higher hydrophobic substituents, so that our log P increases. The negative log P square factors, so that the relationship is a parabolic and there is an optimum value for the log P, beyond which increasing the hydrophobicity causes a decrease in the anesthetic activity. So, with the help of this formula, we can predict the biological activity as how we can change the substituents by changing its physicochemical parameters like partition coefficient. So, that its our biological activity will change. Next is QSAR equations are only applicable to the compound in the same structural class. Same structural class means what? If I am talking about the general anesthetics of the ethers, so that all the compounds that we are taking in our study is from the ethers. So, all the structures are related with each other, like here if I am taking this like phenylacetic acid, if there is a substitution here, so that we can change here X, Cl, Br, iodine, CH3. So, the compound produces from this that this will belongs to the same structural class of the compounds. Now, same structural class, another example of the same structural class like benzodiazepine, same structural class like barbiturate. So, that we can observe the biological activity belongs to the particular class only. Here there are some substituents that the log P value, we can observe the log P value with the help of substituent hydrophobicity constant, and this substituent hydrophobicity constant, it is related with this pi value. So, the substituent hydrophobicity constant is how hydrophobic a substituents related with the hydrogen. So, that all the substituents that we have to take as a as comparison to the hydrogen, we have to compare all the pi value with the hydrogen. So, here what is pH and the what is px? The partition coefficient of the standard compound means it is the hydrogen attached here, and the px, that is the partition coefficient of the compounds with the substituents, like here if the benzene ring and here hydrogen present. So, chlorine. So, ultimately this chlorobenzene have this chlorine different types of pi value. So, that the compounds the hydrophobicity of the compound will change. For example, here we are going to calculate the pi value. Pi value of the chlorine is 0.71 and the pi value of the amide is minus 1.449. So, the pi value is used only for the substituents, hydrophobicity of the substituent. If I am talking about the hydrophobicity of the compounds, then we have to denote with the log P value. Log P, here the log P value of benzene, log P value of benzene is 2.13, log P value of the chlorobenzene is 2.84, the log P values of the benzamide is 0.64. Now, how it comes? So, here we know about the log P value of the benzene is 2.13. So, on the basis of this log P value of the benzene and the pi value of the chlorine, that is 0.71, we can calculate the log P value of this chlorobenzene, because it is the same series of the molecule. So, this chlorine have benzene and this pi value of the chlorine, that is 2.13 plus 0.71, you will get 2.84. So, the log p value of this chlorobenzene is 2.84. So, like this we can calculate the log p value of benzamide. Here benzene is the parent molecule and the substituent present in this CONH2. So, that here 2.13 plus minus 1.49. So, here the log p value is 0.64. Okay. So, the positive value substituents are more hydrophobic, negative value substituents are less hydrophobic than the hydrogen. So, again another example that we can calculate the log p value of the metachlorobenzamide, and the log p value of the metachlorobenzamide, the theoretical value is log p of this benzene, and we have to observe this pi value of CONH2 and the pi value of the chlorine. So, you have to add 2.13 plus 0.71 minus, because the pi value of the amide, that is minus 1.49. So, it will come around 1.35, and the observed value, this is the calculated value and this is the observed value of the log p of the metachlorobenzamide, that is 1.51. So, it is more or less same. So, a QSAR equation may include both P and pi. P and pi means the substituent hydrophobicity constant of the compound of the molecule and the hydrophobicity of the molecule in terms of P value. So, P value measure the importance of molecules overall. If I am searching the molecule against the central nervous system, so we have to observe the P value of the compound, because the P value is directly related with the lipophilicity of the compounds. And the value of P is only valid for the parent structure, it is possible to calculate using pi values. Now, the application, the QSAR equation, which in turn are based on the calculated partition coefficient, can be used to provide the toxicity estimate, by which that we can observe the toxicity also. Now, the calculated partition coefficient are also widely used in the drug discovery, because it is a very important tool that we can observe the biological activity by taking the most important physicochemical parameters involved, and here with the help of this we can observe the molecule, and it is very useful in the drug discovery to optimize the screening of the library and to predict the drug likeness of the designed drug candidate before they are synthesized. So, here in the quantitative structure activity relationship, we can do, if I am going to observe, if I am discovering a new molecule against any type of that, like anti-cancer or the CNS. So, if I am observing the CNS type of drug, so that we can observe the log p value of all the compounds, that is very helpful in identifying the molecule against the central nervous system. This is the references. I have taken this Patrick Gale introduction to the medicinal chemistry. Thank you so much.